Okay, so chapter 3 is all about the cell. And some basic information about the cell is that the definition of a cell is that it's the smallest unit of a living thing, okay? So all living things are made up of these building blocks called cells. Cells are considered to be alive, so even a single cell, one cell is considered to be alive, and there are organisms that are only one cell large, and those would be called our unicellular organisms. And unicellular organisms are going to be microscopic. Okay, so usually you can't see them without a microscope, but they're everywhere around us. They're, living in the water, they're living in the soil, they're even living on your bodies. And probably the one that you're most concerned about would be bacteria. So bacteria are unicellular. They're very, very, very simple living things. Most of them don't cause any problems. So like 99% of them won't ever bother you. They're just living their own lives. However, there are, of course, some that can cause disease. And so the ones that can cause disease are called pathogenic. So they can cause disease. And you'll learn more about them in your microbiology class because there's specific ones that we study and we look at how they cause disease and we look at how we treat it, what kind of drugs might kill them so that we can help animals and humans recover. There are other types of organisms that are not bacteria. So the picture of this big old blob right here, this is called an amoeba. And an amoeba is much larger than a bacteria. It just mostly like lives in water and kind of swims around. Okay, so here's a little picture showing you the different sizes of things. And it's just sort of to show you in comparison. So something like an ant is visible, right? Even frog eggs are visible. So those are probably the largest cells. Eggs are the largest cells. And you can see those with your naked eye, right? But anything smaller than that, you do need a microscope. And we have animal and plant cells, and even some bacteria are visible under the microscope, though many times you can't see them that well. You have to dye them. Otherwise, they're too see-through, they're too clear, and it would just look like kind of like gray blobs. But we do dye them, and then that's one of the ways we can distinguish between different types of bacteria. Now, once you get to the size of a virus is super tiny, so you're not going to really see viruses in regular microscopes. You're going to see them in something called an electron microscope, which regular labs don't have. So even if you return to campus and you're doing, let's say, your microbiology lab, and you're looking at bacteria under your microscope, you can't see those viruses. They're too small. The electron microscopes are really, really intense, heavy, expensive machines that are usually only used in research labs. So it's not something we would have, but we would have pictures of viruses that other people have taken. And then once you get to this range, these guys are no longer alive, right? These are just molecules. So things like proteins, water molecules, those aren't alive. And technically, neither is the virus. So technically, a virus is not alive. And the reason it's not alive is because it doesn't have a cell. So the smallest thing that had a cell was the bacteria over here. Okay, and then this is just showing you what a microscope looks like. So that's just like a typical microscope. Okay, so we're going to start by comparing bacteria cells with a typical animal cell. And so the picture here is a human cell, but just so you know, all animal cells basically look the same. So when we talk about a human liver cell or a human skin cell, your animal's liver cell and your animal skin cell is going to look basically the same at the microscopic level. And what you're going to see is they do have some things in common, okay? So DNA and RNA, those are the molecule of life, and they're going to have your genetic material. And anything that's alive is going to have genetic material. So both the bacteria and the human do have that. The difference is the quantity. So maybe the bacteria has anywhere from like hundreds to thousands of genes, while the human cell has something between like 20 to 30,000 genes. So there's a lot more genes in the human cell. The other thing that they both have is ribosomes, and ribosomes are going to be important for making proteins. So these cells need proteins to survive, and they need a way to make their proteins, and that's going to come from ribosomes. So both bacteria and human have that. And then the other thing they have is a cell membrane. So that's going to be wrapped on the outside. We'll talk about it more in a little bit. And then cytoplasm is sort of this goo stuff. I call it goo, but it's basically like watery material inside of the cell. It's not pure water. There's like proteins and nutrients and electrolytes and all sorts of stuff floating around in there, but it tends to be a little bit thicker than regular water, a little bit more viscous. Any questions so far? One other thing I'll add here is there's some vocabulary associated with this. So bacterial cells are also referred to as prokaryotic cells. And then a human cell would be something called eukaryotic cell. And you can see that the difference is that the prokaryotic cells are very, very, very simple, okay? They do not have a nucleus, no nucleus. While the human cell over here, the eukaryotic cell has a nucleus. That's its nucleus. And then it has all of these organelles. So there's mitochondria, there's Golgi body, there's things like different vacuoles here. So there's a lot of stuff going on, and the bacteria is missing all of that. Okay, and then just really quickly, why are cells so small? So why can't we have like a giant, you know, slime ball cell that could just go around and like eat everything in the environment? So it can't do that, okay? And it has to do with surface area. So my little example for that is, since, you know, you guys like animals, is let's say you had a Great Dane. Okay, so I'm gonna draw a big old Great Dane. So that's my Great Dane. And then you have like a little tiny Chihuahua. Which one of them is going to get cold faster if they're outside, like it's cold outside?
The Chihuahua.
So the Chihuahua is going to get cold way faster, right? And that's because even though the Chihuahua is small, and even though the Chihuahua has less skin than the Great Dane, compared to its body weight, so this is called the surface area ratio. So if you compare the size of its skin compared to the size of its weight, the Chihuahua has more skin to weight. So that means it has more surface area. And having more surface area means you lose heat faster. And in general, this is true. So smaller animals are going to be more susceptible to temperature change than larger animals. Larger animals can maintain their heat better, right? And we're talking about, of course, about mammals that are warm-blooded. This won't be true for your reptiles because they don't generate their own heat anyway. But this is just kind of, again, to show you the difference of surface area. So this actually applies to cells as well. So if you have a really big cell versus a really small cell, the surface area is going to be higher in the small cell, so more surface area. And the surface area is important because things have to go in and out of the cell. So things like carbon dioxide and oxygen, or water, or nutrients, these things have to go in and out of the cell. And they can only go in and out through special openings in the cell membrane. So if you have a really small cell, it can go through very, very quickly. But if you have a large cell, so let's say, kind of imagine like there's doors here, okay? So door, 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 door. Okay, and then this one also has some doors, but it has a lot fewer compared to the size of the cell. So think of it, a good example would be if you had, 10 people in a room with 10 doors, a small room with 10 doors, they can exit that building really quickly. But if you had 10,000 people in an arena, and even though there's a lot more doors, like maybe there's hundreds of doors, 10,000 people, it's really hard for them to get out of that building. It's much more dangerous. So the same idea is going on with the cells here, is that having the smaller cell allows it to let objects in and out much faster. And okay, so some different things about cells. So this is a little bit of extra information. You don't need to know too much about it, but eukaryotic cell, of course, again, is our animals. Okay, so our animal cells belong in this group. And then prokaryotic cells, this one is going to be our bacteria. And then there's one other group here called archaea, the archaea. They look like bacteria, but they're like these really weird things that live in places like hot springs or really, really cold environments or really salty environments. So most of the time we don't worry about them because they're not really causing disease in humans or in animals. It's your regular prokaryotic bacteria that could potentially cause disease. Okay, so let's take a closer look at the inside of this bacteria, and let's just see what like some things are going on. Again, they have DNA, so there's DNA floating around. They have ribosomes. Cytosol is the watery stuff. Okay, and the cytoplasm is just all of that together. It's just a combination of all of those things floating around in that watery goo. In addition to that, though, they're going to have cell membrane because all bacteria have cell membranes, and they're going to have a cell wall, and this is going to be important. So we're going to look at cell wall more in a little bit of detail. And then many of them have a flagellum, not all, okay, so only some bacteria will have this. An example would be E. coli. Have you guys heard of E. coli or maybe like a disease caused by E. coli?
Yeah.
Do you know like what it does to you?
Isn't it kind of like food poisoning?
Yeah, and that's basically the symptoms you would get. So E. coli is a bacteria that lives inside of the gut of many animals. And in general, if something lives inside of your gut, it means that it's going to come out in your feces. And this is the reason why they tell you, don't touch feces. Or if you go to the bathroom, make sure you wash your hands. Because if you get any fecal matter on your fingers, on your hands, there's probably going to be bacteria in there. And E. coli is one of the types of bacteria. So if you're handling animals and you touch their butt area and there's some fecal matter there, you could get E. coli on your hands. So you can catch illness that way. What happens with food poisoning is the idea is that maybe the food got contaminated with E. coli, so maybe somebody wasn't washing their hands properly when they were handling that food, or during the processing of the food. So like maybe they were butchering a cow and some of the E. coli from the cow's gut got into the meat. So then the meat could potentially have it as well. And with raw meat, it means there's bacteria potentially in that raw meat. If you cook it, you're usually fine. But if you're not cooking it well enough, you're not cooking your hamburger or your steak well enough, there could still be some E. coli, and then that can cause food poisoning. So anyway, the reason I gave that example is because E. coli does have flagella, so E. coli is able to swim around. Not all bacteria can swim around. Okay, oops, I lost my little arrow. There it is, okay. So next, let's look at this animal cell. And you're gonna see this animal cell is way more complicated. So yes, there's DNA. The DNA is actually in here. Okay, so that's gonna be your DNA. But the DNA is inside the nucleus. And the bacteria cell did not have that. We do have ribosomes, so that's something we have in common. But then we have all this other stuff, lysosome, Golgi apparatus, mitochondria, centriole. These are all special things inside the animal cell that the bacteria cell didn't have. However, if you look closely, what's something that the bacteria cell had that the animal cell does not have? Here, I'll go back to the picture. So look at this picture. What's something that this guy has that our cell doesn't have?
Cell wall.
The cell wall. Okay, the cell wall. That's a big deal. The fact that animal cells don't have a cell wall means that it's an easy way to distinguish between bacteria and animal cells. And so why do we care? We care because this is going to be the target of many drugs. Target of antibiotics. So when you take antibiotics, what you're trying to do is you're trying to take a drug into the body that will kill those bacteria, but will not hurt your animal cells. And this will work because if the antibiotic targets the cell wall, it destroys the cell wall and the little bacteria basically pops open and dies. But because the animal cell has no cell wall, no problem. If you target a different part of your cell, like let's say you target DNA, there are drugs that target DNA. The problem with that is that both the animal and the bacteria have DNA. So there's gonna be some side effects. And you're gonna see that with drugs when you learn about them more, is that certain drugs have more side effects than other drugs because there's more similarities between the human or animal cell and the target cell. What else? Is there something else here that you see that you don't really see in the animal?
Are the little things poking out from the cell wall, is that still the flagellum or is that something else?
These are called pili, and they're actually there for attachment, but that's a good catch. So the pili are for attachment purposes, and a way to think about it is, again, that E. coli is living inside of your gut. So how does it make sure that it doesn't get pushed out with your feces all the time? So what they do is they stick to your intestinal wall, to your cells, using pili. It's like an attachment thing. And then they can also use it to stick to each other. It's pretty cool. They can actually do this weird thing where they exchange some genetic material. So one E. coli can find another E. coli and they stick together and they can exchange some genetic material. So that's true. This is pili are not really found in animal cells. And then is there any other really big long thing? That looks different there.
The flagellum?
Yeah. So here's what's interesting. Flagellum can exist in animal cells, but they're very rare, and that's because they're used for swimming. Okay, so the flagellum is used to swim. Now, when you have a body cell, like let's say you have a liver cell, you don't want that liver cell swimming around to a different part of your body. Like it needs to stay put. So in general, most of your cells do not have a flagellum. Skin cells don't have it, you know, liver cells don't have it, bone cells don't have it. The only cell in the animal that's going to have a flagellum is the sperm cell. Okay, because the sperm cell does need to swim. So for that reason, it actually just has one flagellum, a single one. In comparison, you can see that bacteria sometimes have many. So sometimes they only have one, sometimes they have many. It depends on the species. Okay, any questions so far?
So with like things like tadpoles, would that be like a flagellum, or is that like part of their whole like process of growth?
So the tadpole does have a tail. The difference between a flagellum and a tail is that we're looking at remember this is microscopic. So it's so tiny. So one single
Okay.
little tiny cell, if you looked under a microscope, a sperm cell looks like that. Okay, so it's
Mm-hmm.
literally one cell with a tiny tail. The tail of a tadpole actually has millions and millions of cells because the tadpole is an animal. So the tadpole, by the time you can physically see it with your eye, it has so many cells. And if you looked in
Okay.
there, and it's not just one type of cell either, like it would have skin cells, muscle cells, bone cells, blood cells, like all sorts of cells. But the purpose is the same. The purpose is to swim.
Okay.
Okay, so very briefly, I'll talk about plant, but just very briefly, because again, as a vet tech, you don't really care about plants. And in general, they're not gonna be like part of your medical background, but we just wanna compare plants to animals. So what we really wanna look at is what is some differences, how is it different from an animal? And to figure that out, again, let's look at this picture here. So this is your typical animal cell. And then here is your typical plant cell. So they do have some stuff in common, okay? They both have the nucleus. They definitely have ribosomes again. They have that cytoplasm again. What are some of the things? We talked about cell membrane. So they do have some stuff in common, but is there anything really obvious to you that's different?
It's more square and
It
it's
is more
not
square.
round.
Yeah, it has a different shape. Animal cells in general tend to be round-ish, though there are actually different shapes of animal cells as well. But the shape of the plant cell comes from a cell wall. So they do have a cell wall, unlike the animal cell, but just so you know, their cell wall is different from bacteria. So they're not the same. They're made out of different materials, but they have the same purpose, which is its protection, right? It's a protective layer around the cell, and it does give it its shape, you're correct. So it gives it kind of a box-looking shape. And then when you have a bunch of plant cells together, they tend to look like little bricks. And that's part of what makes the plant strong, because it doesn't have bones, right? Plants don't have bones. Like, how are they able to stand upright? They have these cell walls, and then little bricks stick together to form their tissues. Anything else really obvious that's different? I think I kind of heard something, but it wasn't very loud. Central vacuole. There you go. Good. So if you look back at this picture, let's see if they even have vacuole in this picture. I don't think they labeled it, but there are little tiny vacuoles inside of an animal. So there are vacuoles, but they are really small, while this one has a really large one. Okay, so it has a large central vacuole, and the purpose is that this is where a lot of the water is going to be stored. So plants, for any of you who maybe own a plant, you'll notice if you don't water it for a while, so you had your nice plant, this is your nice plant, and then if you don't water it for a while, sorry, that looks like a hand. Okay, let me try something else. So this is your nice plant, happy plant. If you don't water it for a while, it tends to like droop and everything kind of looks like wilted and sad. Okay, so when it's wilted, it means that the central vacuole has lost water. And it just means you haven't watered in a while. But a lot of times if you have a wilted plant and you give it some extra water, it will look nice and happy like this one. So this one has lots of water. And it's just storing the water for later, that's all, okay? And then the water also provides it with some structure. Okay, and then the last thing I'm gonna point out, of course, is the chloroplast. So chloroplast is unique to our plant cells. It is not found in animal cells, and that's because its purpose is photosynthesis. Okay, so photosynthesis using the energy of the sun, that's something plants can do, but no animals do that. It also is what gives the plant the green color. And that's all you need to know about plants. So just know that they are different from animal cells. They have cell wall, central vacuole, chloroplast, and those are different from our cells. Okay, I'll go back to endosymbiosis later. I'm not sure how much you need to know about that. So now let's look in a little bit more detail. Inside of the cell, we have organelles, okay? And organelles basically have their own job. Each organelle has a job, okay? And its job is very specific, and then it will help the cell to survive. So for example, some might be involved in digestion, some might be involved in making proteins, some of them have to do with energy. So it just depends on which organelle we're looking at. Okay, so the first thing they're going to talk about is the cell membrane. Now, cell membrane is found in all cells. Okay, so cell membrane in all of them. The cell membrane of the animal cell is this thing right here. The cell membrane in the bacteria is this like thin one on the inside. So it's on the inside and then the wall is on the outside. And then in the plant also, there's a very thin cell membrane here and then the wall is on the outside. But every single cell has it. So what does it do? It's made out of phospholipids. So last week we talked about that there are four life molecules, and they were proteins, carbs, right, carbohydrates, lipids, and then nucleic acids. So those were our four life molecules. What we're going to look at is that lipids can help form cell membranes, and the type of lipid is called a phospholipid. So basically, on the left is the really complicated way that it looks, like all the carbons and hydrogens inside of it, but we're going to simplify it and draw it as a head with little two tails. And it's not by itself, okay? So the phospholipids, there's a bunch of them next to each other. They kind of line up like that. And in addition to the ones that are lined up like this, there's going to be another layer that's facing downwards. And I'll show you some better pictures of my drawing. Okay, so these are individual phospholipids that are all lining up, okay? And so then this will be the outside of the cell, and this will be the inside of the cell, and this part is the actual membrane, the membrane that is separating the outside of the cell and the inside of the cell. So let's see what that looks like. Well, we'll go back to that. So this is what it looks like. So notice this is, of course, a much more beautiful drawing than mine, but you can see we have the heads, we have the tails, and then it forms a bubble, okay? So the simplest idea of a cell is that it's basically a little bubble with water on the inside and water on the outside. Now, why is it able to separate the water? It's able to separate the water because of the way that the phospholipid molecule works. So when you have the head with the two tails, some things you should know. The head is considered to be hydrophilic. So hydrophilic means that it loves water. But the tails, so it has two fatty acid tails. The tails are hydrophobic. And you've probably heard the term phobia before, right? It means to fear something. So hydrophobia means it is afraid of water. That means it's going to stick its little tail away from the water. And so that's what's happening, is that the heads are always facing the water. These heads are facing the water inside of the cell, and these heads are facing the water outside of the cell. But the tails always face inwards. And that's going to be important for cell transport, which we'll get to next week. That's a different chapter. But just understand, this is their arrangement. Heads are always facing towards the water, tails always facing away from the water. The other thing is that the name for this structure is a phospholipid bilayer. And so it's called a bilayer because there's one layer here and a second layer here. So there's two layers, that makes a bilayer. Any questions about that? All right. So continuing on, just so you know, it is a little bit more complicated than that. So in the picture, it just shows you the phospholipids. It doesn't show you anything else. But in a real cell membrane, you have other stuff too. So yes, you have the bilayer. Okay, so you have the bilayer, but you also have proteins inside of the bilayer floating around. So they're showing you here, there's the head, there's the tail, there's the head, there's the tail, there's the head, there's the tail. Oh, but look, we have a protein here. And what this protein here is, for example, this is a channel protein. And so a channel allows things to cross. And that's going to be important because other things are not allowed to go through the membrane. In fact, the name of the membrane, another name for it, is that it's semi-permeable, semi-permeable membrane. So what that means is if something is just trying to get into the cell, it's like coming, boop, and it's going to bounce off. So it doesn't get to go inside of the cell. However, if it uses a channel, depending on the type of molecule, it might be allowed in. So it just depends on, we'll go into detail about that. Some things are allowed in, some things are not allowed in. And then you have other types of molecules as well. So for example, this one, look, notice it doesn't look like a channel, right? There's nothing going through it, but it has this thing, some sugar stuff sticking off of it. So this would be basically a type of recognition protein. So another name for that is a marker protein. And what it does is it helps you identify your cell. So for example, your liver cells will have special markers letting your body know that that's a liver cell. And then there'll be other markers on your pancreas, and there'll be other markers on your heart cells. So this way your body can recognize different parts of itself. It's also related to blood type. So maybe you've heard of blood type before. Do you guys know anything about blood transfusions?
The two patients doing the transfusion would have the same blood type.
Yeah, we need them to match. And actually in animals, it's way more complex than in humans because you have to deal not just with blood types, but with different species, right? So you can't like give cat blood to dog blood, like that doesn't work. But even within dogs, there's different blood types. So you have to make sure you match. The same thing's true for humans. Okay, so maybe some of you know your blood type, like maybe you know, oh, I'm blood type A. So if you're blood type A and you have an injury and you need to get blood, you can get from another person who's blood type A, but you could not get it from someone who's blood type B. And what does that mean? Like, where does it come from? It's because of these special markers on your blood that tell you what type you are. So if you're type A, you have a special marker that means A, while the blood type B person has a different marker that means B, and the two do not match each other. So you're gonna have a really bad reaction if you try to give the wrong type of blood. And I'm sure you'll learn more as a vet tech, like how to do blood transfusions, but basically the blood packets that you receive, they will be labeled already with the correct type, so you'll know what type they are. And then you would have to do like a blood test on the animal to find out what type they are. And then you make sure that they match. Okay, cell wall, we're gonna skip because we don't really care that much. The only really interesting thing about this is that the cell wall of the plant is made up out of cellulose. So cellulose is a type of sugar, but it's a type of sugar that we as a human, for example, cannot digest, right? So humans can't digest it, but some animals can. Can you think of some animals that can digest cellulose? So like, who eats grass? Lots of grass.
Cow.
Cows, okay. So cows can digest grass, but you know what? It's actually really hard to do. And the reason they can do it is because they have special bacteria in their gut, special gut bacteria that help them to digest cellulose. It's also why they like chew their cud for so long, right? Like they chew the grass so long. And they also have special stomach compartments. I'm sure you'll learn about that more, like the ruminant and like how does that work. So it's really, really hard to digest cellulose. And so it takes a lot of special processes. If as a human, you ever tried to eat grass, nothing would happen, okay? So you would get zero energy out of it. It would not be digestible and it would just come out in your feces. And that would be the end of that. Okay, so let's continue. That was basically the cell membrane. So we just talked about that. So now let's look at things that have to do with proteins. So what are some things that have to do with proteins? So I'm going to do a little drawing for you because I think it's easier to first draw it out and then let's look at their more complicated picture because their picture is really, really detailed. So here is your typical cell. So that's going to be its cell membrane. And inside of the cell, you typically have a round compartment that looks kind of like that. So right now it looks like a little eyeball, right? And this whole thing is called the nucleus. But the nucleus has some special parts. The darkest part in the center of the nucleus is called a nucleolus. And then the layer around the nucleus is specifically the nuclear membrane. Okay, so what's going on in there? In general, if they ask you, like, what does the nucleus do? There's a couple answers. You can say that it helps direct the cell. It helps control protein synthesis, stuff like that. How does it do that? Well, it has DNA inside of it, okay? So there should be DNA molecules inside of the nucleus. And the DNA, of course, holds the genes. Outside of the nucleus, you're going to have some special other structures. So I'm just going to draw them really, really simply here. What these wiggles mean is that they're made out of membranes, okay? So it's similar to the cell membrane and the nuclear membrane. They are made out of membranes, but they have special jobs. So this one we're going to call rough ER, and then the blue one we're going to call smooth ER. And the ER stands for endoplasmic reticulum, which I'm not going to spell out right now, but you'll see it in the slides coming up. So endoplasmic reticulum is just the name for these membranes. So what's the difference between the smooth and the rough? Well, the rough actually has little rough particles on them. So if you see it underneath the microscope, it looks rough, while the smooth one does not have these particles, and that's why it looks smooth. What are those particles? Those are ribosomes. Okay, and the purpose of the ribosome is to make protein. But the question is, well, how does it do that? Like, how does it know how to make a protein? The information on how to make a protein is inside of the genes. So the genes has the info on the protein. Like, how do I make this protein? But we're going to need some worker molecules. And the worker molecules, let's see, what other color have I not used? Let's use orange. The worker molecules are much smaller. And those worker molecules are going to be called RNA. So think of these as the workers. So basically the DNA has information on how to make the protein. The RNA is going to copy that message. It's going to copy how to make the protein, and it's going to exit the nucleus. And it's going to come over here and find itself a ribosome. And it's going to attach to the ribosome, and the ribosome is going to help actually make the protein. So an important point here, this is why I'm kind of drawing it out for you, is that the DNA is actually stuck inside the nucleus. It's stuck inside because it's a really, really large molecule, never leaves the nucleus. But these little ribosomes, sorry, not ribosomes, these are RNA. The little RNA, they can leave through special pores because they're much smaller. So those are pores. They're just special little openings. And then the RNA is able to leave, it's able to go out into the cytoplasm and find itself a ribosome, and then the ribosome will make proteins. So that's kind of the big picture. Now let's look at what your book says. So see, you have this stuff in the book, but it's like way more complicated looking. So let's look at it step by step. So here was the DNA molecule, okay? Here's your mRNA molecule. So mRNA is just a special name for RNA. We'll talk about that more in the future, like why it's called mRNA. But the mRNA is going to leave, and see this little gap here. So that little gap is the pore. And when it left, it found itself a ribosome. So there's the ribosome. And the membrane that the ribosome is on top of is called the rough endoplasmic reticulum. Okay, so this picture basically matches what I just drew over here. How do we feel about that so far? Any questions, or are we okay so far?
I'm okay.
Okay. So once the RNA is there, it's going to start to help build a protein, and let's make the protein yellow. Okay. So it's going to start building, can you guys see that yellow? It's kind of hard to see, but it's going to start building that protein. So the protein is going to be in yellow. Well, the protein needs to go somewhere. Sometimes the protein stays within the cell itself. Okay, so let's say, for example, this was a blood cell, like a red blood cell. Red blood cells have to make a lot of hemoglobin. Hemoglobin is a type of protein. And so they are full of hemoglobin, which means that they are making tons and tons and tons of hemoglobin inside of their cells. That's before they become mature. It's a whole long process, but basically they're making like millions of hemoglobin until they are ready to be mature, and then they're going to carry oxygen for you. Other cells have different jobs. So let's say you need to make insulin. That was one of your proteins that you could look up in the discussion. So insulin is a special protein that needs to be released after digesting food. So I just ate a meal, I need some insulin. How am I going to get it? Well, you're going to make the insulin protein over here. Okay, so you're going to make it next to all of these ribosomes. But once I make it, I need it to leave the cell. So what's going to happen is they're going to be packaged into these little bubbles by the rough ER. So the rough ER can basically transport these proteins by wrapping them in little bubbles. Once it wraps them in the bubbles, it's going to take it to another structure over here called the Golgi body. Okay, so that's the Golgi body. And what will happen is, so we're assuming that these guys are filled with protein. Once they reach the Golgi body, they're going to fuse, and they're going to give the protein over to the Golgi body. And now the Golgi body is going to modify these proteins. And once it's modified them, it's going to wrap them in its own bubble. So that happens a lot. You're going to see a lot of little bubbles. So it's going to wrap it in its own bubbles, and then it's going to send it out of the cell. So what happens is, here's your protein, going to be wrapped in this new bubble. Once it fuses with the cell membrane, it can actually exit the cell. Okay, so now the protein has been released. So these are the proteins have been released by the cell. So what happens with insulin specifically is that the insulin is produced inside of your pancreas. There's special cells that make it. So it has DNA with genes on how to make insulin. It has RNA to help build the insulin. It has the ribosomes that actually creates the insulin. Then the insulin is going to be transported by the rough ER. It's going to be taken over to the Golgi body. The Golgi body is going to modify it, and then it is going to send it out of the cell. Send out of cell. Once it's out of the cell, it basically goes into your bloodstream, okay? And then once the insulin is in your bloodstream, it will go wherever it needs to go. So now let's look at this picture again. So we already had our rough endoplasmic reticulum. And then what they're showing you is that the molecules are traveling over here to this structure. Here's your Golgi apparatus. And there's your proteins again in the Golgi apparatus. We're putting them in a special little bubble that's just called a vesicle. That bubble comes over here, and now the proteins have been released. Okay, so they are released out of the cell. Now, I think the example here, it says to milk ducts, so that means that they're showing you how to make protein for milk, like breast milk. But every cell has the same general purpose, which is to make proteins, and then which type of protein it makes depends on the cell type. So if you're looking at mammary glands would make the proteins that go into the milk. If you're looking at pancreas cells, some of them make insulin. So any questions at all about that process? All right, so that's fine. I think that basically covers that whole section. This is just a close-up view of the nucleus, so just be careful. This is not a cell. This is not a cell. It is just a nucleus. And they're just showing you the different parts of the nucleus. So there's the dark part here called the nucleolus. There's the nuclear envelope, which is the same thing as a nuclear membrane. And then these are the pores, which are the special little openings. And things like RNA can exit, but not DNA. DNA is too big. Okay, so then we go back to that picture again. This is showing you a close-up of what actually happens at the ribosome. Okay, so this is a ribosome close-up. So this thing right here is the ribosome. And what they're showing you is the mRNA molecule, which was the little worker, and it's showing you the protein that is being built. So it's just showing you in a little bit more detail of what that looks like. Okay, and then let's continue. So this whole system, it's just called, I think it gave you the vocabulary over here, it's just called the endomembrane system. That's just telling you that because there were so many membranes involved, right? There was a rough-ER membrane, there was a Golgi body membrane, so there's just different membranes involved. The other thing that if I go all the way back to my picture that I drew, notice that the smooth ER had nothing to do with protein production. What the smooth ER does is it can help build lipids. So it can make lipids for you. And then lipids are necessary to form these little bubbles and to form cell membranes. So lipids are important. The other cool thing it does is it can detoxify. So it can take harmful chemicals and reduce them to non-harmful chemicals. And that's important so that the cell can survive. All right, continuing on. The name of the little bubbles is just called a vesicle. So you'll see vesicles in different places, like this is just called a transport vesicle. And then there's also vesicles like when neurons signal, you'll learn about that probably in anatomy, they have their own vesicles. Okay, next part, new topic is cellular digestion. So I don't want to redraw everything that I drew in my cell, but just like really quickly, we had a nucleus, we had some smooth ER, we had some rough ER, blah, 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 and we had the bubbles, and then we had a Golgi body. Okay, so that's kind of like where we left off. So there are other organelles in the body, and so I'm just going to summarize some of them. These guys you've probably heard of before, and they're called mitochondria. So who knows what mitochondria do?
It's like the powerhouse. It's the it kind of helps to work the cell.
Yeah, so that's usually the term people use, the powerhouse of the cell. So when you think of powerhouse, like you're trying to get some power, what is it that you're looking for? You're looking for some energy. Okay, so basically all of the stuff that the cell did so far, like it was making proteins and it was sending them here and there and outside of the cell, all of that requires energy. It can't just do it without energy. So where is it getting the energy from? It's getting it from a special little organelle called the mitochondria. And the mitochondria, the actual molecule it forms is something called ATP. So you'll see ATP more in the future, but it forms ATP in a special process called cellular respiration. And we have an entire chapter on that, on how that works, but that's its job. Now, outside of that, we're gonna have some other things with jobs. So we're gonna have, let's see, these little dudes. Again, they're just gonna look like bubbles, which is why it's kind of confusing. But we have something called lysosomes. And lysosomes have digestive enzymes. And they can be used for several things. They might be used to break down large molecules. They're used, for example, in white blood cells to destroy bacteria. So the lysosome can actually digest bacteria and destroy it so it's not harmful to you. It can also be used to digest old parts of the cell. So let's say there's like a non-functioning mitochondria, it's too old, it's broken, it doesn't work anymore. The lysosome could actually envelop it and digest it and break it up into small pieces and then recycle it so that it can be used for something new. So lysosomes have that job. We also have little bubbles that are just called storage vacuoles. So vacuoles are used to store things. It might store water, it might store other molecules. In the plant, we had that really big central vacuole that was used for also for structure, right, to help support the plant. But in the animal cell, they're just like little tiny bubbles, and they'll be all over the cell. They'll be everywhere. Let's see, what else do we have? So we did Golgi body, we did mitochondria. We have something called a cytoskeleton, which is a little bit confusing in terms of what it looks like, but this is kind of just how I'm going to draw it. So cytoskeleton provides structure. And you might wonder, well, what do you mean structure? So not all cells are round like this cell. A cell can have different shapes. So let's say we had a cell such as a neuron. Okay, so I'm going to draw a neuron for you. A neuron has special projections off of it that kind of look like this. And then it's like really long, and then it has more projections. So you might wonder, well, how was it able, and there's this nucleus, how was it able to keep this shape? It was able to keep the shape because special cytoskeleton molecules form that shape, okay? So inside of it, it will create a certain shape, and then the cell membrane will wrap around it, and then now it has the projections that you want. So it is used for structure, and they're actually types of protein fibers that do this. Okay, so let's just check what we have in the next couple slides and make sure that we answered everything. So lysosomes, yes, here's your lysosome. Again, they just look like a little bubble, but their main purpose is that they can break down things. So here they said it's used for digestion, not the same kind of digestion as what you're thinking of. So when you eat food and digest it through your digestive system, that's different. That's at a much larger level. We're talking about at a very microscopic level where a particle came into the cell and needed to be digested, or like I said, let's say you have a damaged mitochondrion right here. So it's damaged, we don't want it anymore. So it's gonna fuse with the lysosome, and it's gonna just break it down into little pieces that's no longer necessary. And then either it can spit it out as debris, like it can kind of throw it out into the blood, like outside of the cell, and then you just get rid of it with your kidneys, or you might recycle it. And then vacuoles, again, are just used for storage. Peroxisomes are another little type of bubble. They're a special type of bubble that has special enzymes. And what they do is they break down some toxic molecules. Okay, and then we did talk about mitochondria. So this is what a mitochondria really looks like. It's like a lot cooler than my drawing, but my little drawing was just a very simplified kind of bubble like that. And its purpose, again, is to make energy. Oops, and then actually this slide talks about it. The name for the process is cellular respiration right there. Chloroplasts, so chloroplasts are only found, again, and remember they're in plants, not in animals. Plants, not animals. And their purpose is the opposite. They actually have the opposite purpose of a mitochondria. Because they don't produce energy, they use energy to make sugar. And then mitochondria use sugar to make energy. And then here's cytoskeleton. So cytoskeleton is made out of special protein molecules. Oh, okay, good. So the last couple things here are the cilia and the flagella. So I'll go back and I'll add that to my drawing. And anything else? Communication, we're not gonna worry about that right now. I'm not gonna worry about specialization either. Okay, so let's go back to my drawing. And I'm gonna add some things for you that I think you'll find useful. Oops, I didn't mean to erase the whole thing. So let me draw the cell membrane again. But I'm gonna draw some extra stuff for you that you'll find useful. So some cells will have these special projections, and then some will have a really longer thing, and then some will have a really, 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 really long thing. Okay, so the really, really long thing is the flagellum, right? And remember that in animal cells, most animal cells don't really have this. It's really only the sperm that would have it, but it's used for swimming. However, some will have a much smaller structure called cilia, and that's more common. So cilia can also be used to move. They basically wiggle. However, the way our body uses it is actually really cool, is you have cilia in places of your body where there's also mucus. And you basically move the mucus. So for example, in your nasal cavity is producing mucus. So that's behind your nose, right? And what it's trying to do is it's trying to trap particles that are trying to get into your body. And sorry, I forgot an O there, mucus. So what's happening, actually, I didn't spell that right. Actually, I think you can spell it without the O, but oh well. Okay, so mucus What it's doing is it's trapping little particles like dust particles or pollen particles that are trying to go into your airway, and then the cilia will sweep the mucus with the trapped particles out of your nose. And so you're going to start having like a runny nose that's dripping out. Another place where you have cilia that's pretty cool is in the fallopian tube of the female's reproductive system. So in the fallopian tube, the egg is trying to move towards the uterus, and the egg does not have a flagellum. So sperm cells have a flagellum and they can swim towards the egg, but how can the egg move towards the sperm? Like, it doesn't have a way to swim. So what the cilia do is they sweep the egg towards the uterus and allow it to move that way. They provide a current. Then the other thing I showed here, these are called microvilli. And microvilli are different than the cilia and the flagellum. So the cilia and the flagellum, their purpose was to move. Microvilli do not move. Their purpose is simply to increase surface area. So remember how we talked about how cells can't be too large, they can't be too large, so smaller cells are better? Well, sometimes if a cell is getting kind of large, but it wants more surface area, it can create these microvilli. And so the idea is a good place where you will find this is in your intestines. And what will happen is, let's say food particles were trying to enter the cell. Okay, so if these were little food particles and they were trying to enter the cell, there's only so much space on the cell membrane. But if you form microvilli, now you have a lot more surface area and a lot more place where the food particles can actually enter into your cell. So you're going to see microvilli in the intestines, and you're also going to see it in some other places where transport of materials is really, really important. Any questions about any of that? Okay, so I'll just double check and make sure we covered all the information in these slides, but I think we're pretty much wrapping it up. So we just talked about cilia versus flagella. And again, this is a sperm cell here that they're showing you. And then these little cilia are probably inside of your respiratory tract. So that can be your nasal cavity, it can be your trachea, all of those have cilia. So this is just showing you, the last couple slides are just showing you different things that your cells might do. So for example, one type of cell in the body that's used for communication is the neuron. It's a special type of cell. And then there's other types of special cells. You might have so nerve cells are special. You might have muscle cells. Muscle cells are for movement. You might have skin cells. You might have liver cells. So each cell has its own job. And then they're also showing you some plants here. So this is a plant cell. This is a plant right here, like a leaf. And then there's root cells and so forth. Okay, so that concludes today's lesson. Any last questions?